An endocrine cell sends a signal molecule, or ligand, towards the bloodstream, where a target cell with the correct receptor may interact with the ligand in order to start the reception process.
The G protein attached to the membrane acts as a switch. When GDP is bound to it, it is inactive. Once the signal enters the active site of the receptor, the G protein interacts with the receptor protein, and the GDP leaves the G protein, causing an interaction with GTP to be favorable, resulting in an activated G protein. The G protein leaves the receptor and binds to the enzyme, activating it and allowing it to conduct a cellular response. The GTP used to activate the inactive enzyme is converted to GTP and a phosphate is released. The activated enzyme splits IP2 into DAG and IP3, which is a second messenger. The IP3 then goes to the ion channel and interacts with it. This causes the ion channel to open its gate and release the calcium. Calcium is also a second messenger, which starts the process of cellular response. A second messenger from the cytosol, in this case calcium, activates an inactive protein kinase 1 and therefore becomes active protein kinase 1. Then the active protein kinase 1 binds to ATP and an inactive protein kinase 2. It then releases ADP and active protein kinase 2, which is binded to the phosphate. This then occurs again, and active protein kinase 2 binds to ATP and an inactive protein kinase 3, in which ADP and an active protein kinase 3 with a phosphate is released. Then the active protein kinase 3 then binds to ATP and an inactive protein. Then ADP and an active protein with a phosphate is released. After each of these occur, a phosphatase takes a phosphate off, which then inactivates the active protein kinases and the active proteins. This then allows for the process to occur again as they are inactive.
A phosphorylation cascade is done in transduction rather than just a few steps in order to more easily amplify and regulate the process. More steps in a cascade result in much more opportunities to regulate. With a few number of steps, it is much harder to find a way to change a response in an instant rather than doing it as a process. For example, you may gradually slow the response by inhibiting multiple enzymes along the way. The phosphorylation cascade then leads into the response. One possible response that can result from a cell signal is enzymatic activity. The activity of a particular enzyme may be activated so that it can carry out all of its functions in the cell.